Okay, greetings. This is a lab that's going to be focused on the male reproductive system. This would be in your lab exercise book, exercise 31. And the objectives for this lab is to review the gross anatomy of the male reproductive system, including any model images. I may include model images as attachments, but also take a look at the images that are in your book, okay? And I'm also going to include the images where I can in this PowerPoint. So it's going to be helpful to review the PowerPoint and follow along with your book opened up. Review the microanatomy of the testes and sperm development, including review the micrographs, and complete the worksheet. Pages 563, 564, upload the worksheets by Friday, 5 p.m. Now, if you go into the week that we're in now, and if you go into the section on lab, I'll have the link. This is going to be put up on the YouTube, and I will also have a document that is in PDF form. So when you unload it, it comes out like a form, F-O-R-M. In other words, you fill in all of the blanks that's going to be for the worksheet for pages 563, 564. Complete it, make sure you put your name in it, and then you can upload it in that same site where you downloaded, and we'll give you the grade for that. How's that? So let's move on. First off, what we're going to be doing is examining the gross anatomy of the male reproductive system, the testes and the accessory ducts. Now, the primary sex organ is the testes. So what are the accessory ducts and glands? This includes the epididymis, the ductus deferens. It also goes by the name vas deferens, the ejaculatory ducts, urethra, the seminal glands, prostate gland, and bulbourethral gland. That's a mouthful, but these are very important glands that allow for the successful transfer of sperm. And I'll explain more of it as we move on. We'll also be noting the external genitalia. Now, the function of the male reproductive system is A, produce male sex hormones, B, produce and store male sperm cells. These are referred to also as gametes. Okay, any reproductive cells, whether it's ovum or sperm, are referred to as gametes. They are also to transport the sperm and supporting fluids to the female reproductive tract during sexual intercourse. Now, the development of the testes. This is where we begin in the first activity section. You're going to note that the development occurs from embryonic tissue close to the kidneys. And as the embryo moving into the development of the fetus, during this development, these testes will descend toward the inferior position of the body cavity. Around the 7th month, the testes travel through the inguinal canals. These are passageways from the abdominal cavity to the scrotum. And the final descent into the scrotum occurs near the time of birth. Now, the testes has attached to it the ductus deferens, and what you have also are blood vessels, nerves, lymphatics, and a spermatic cord. All these structures are enclosed by connective tissue sheath and also by some smooth muscle. So if you take a look at the second image there, you'll see the entire breakdown, cross-sectional view of the testes. What you're going to find also are serous membrane, which is continuous with the peritoneum, and lines the walls of the testes and scrotal sac. The scrotal cavity surrounds the testes and is lined by serous membrane. And then you have the tunica albuginea, and that is a fibrous capsule covering the testes. Now, you're going to see also a site called the rete testis. This is a site where the seminiferous tubules converge. They will form sort of this short tubular network. This network transports sperm through the short ducts leading to the epididymis, okay? And so you can see here, 2 months after conception, you have the developing testes. Notice that it's somewhat retroperitoneal and will eventually have this connective tissue band that will lead it eventually into that inguinal canal. So you move from 2 to 3 months and you start seeing the descent occurring and by birth you have the descent into the scrotum. If you'll notice here in this diagram, you have, of course, a cremaster muscle, a dartos muscle, which we'll talk about in a little bit. This would be the entire area of the scrotum. What you notice along this spermatic cord is nerves, artery, a venous plexus, and then the ductus deferens. Now, the ductus deferens is absolutely essential as a transfer canal. You have what they call the superficial inguinal ring. Now, notice that this is somewhat an opening. This is actually an area where someone might have, they might call it a hernia, where some, due to abdominal pressure or excessive weight, etc., some of the intestinal will actually protrude in through this area here. And it has to be then pushed back up and closed off, okay? Now, you also will notice over the cap, the top here, this is the ependymus. We're going to get more into that in a bit. If you look here, here's the ependymus, or excuse me, the epididymis, I'm sorry. The epididymis, you have this structure that is this arete here, okay? You have the seminiferous tubules, they deliver sperm up into the structure, and then there's a couple of short canals that will deliver the sperm into the storage area, the epididymis, okay? You'll notice the layers in the scrotum, the skin, the dartos muscle, the superficial fascia. Then underneath that is the cremaster muscle, which we'll get into in a bit. And then you have this area of space between the actual testes itself and the cremaster muscle, and that's called the scrotal cavity. Here is, as they were mentioning, the rete testes here. You have basically the septa testes, basically the separations for the seminiferous tubules. You have also this outer tough layer, this tunica albuginea, and then you also have an outer layer here around the epididymis, the tunica vaginalis. Okay? Now, accessory ducts. Sorry about mispronouncing a few things earlier. I apologize for that. They transport sperm and glandular secretions to the outside. So the epididymis is a highly coiled tube. It starts at the superior margin of the testes and then kind of moves down, but eventually connects up to the ductus deferens, otherwise known as the vas deferens. The vas deferens starts where the epididymis is attached to the inferior part of the testes. Now you have what is called the ejaculatory duct. This is formed by the union of the ductus deferens and the duct of the seminal gland. At the urethra, this is the next area. This is the structure that conveys both urine and sperm to the outside. Keep in mind that urine is going to have a much lower acidity, and you have to protect the sperm from the damage from the acidity. We'll get into that in a little bit. Now, the urethra has several different parts. The prostatic urethra begins in the internal urethral orifice in the bladder wall and passes through the prostate gland. So it's called the prostatic urethra. The membranous urethra passes through the muscular urogenital diaphragm. And then we have the penile urethra, which passes through the shaft of the penis, ends at the external urethral orifice. And you can see this pathway here. Here we have sperm stored in the epididymis. You then have the transfer following along this ductus deferens, otherwise known as the vas deferens, and you come up into this area where we start having the seminal gland, the prostate, and the bulbourethral gland. We're going to talk about those in a few minutes. But what happens is that you have this change going over to what would be the ejaculatory duct. As part of it passes through the prostate, joins together with the urinary urethra. Okay, this is part of the prostatic urethra, they also call it. This is bringing urine. This is bringing sperm or semen. And what you're going to have is they meet together, and that's where you start having the other parts of the urethra. You're going to have the penile urethra right through here. Okay? Now, notice the positions of, for example, the pubic syphus, the urinary bladder, and basically how the penis eventually goes deeper into the body structure here. Now, when we examine the gross anatomy of the male reproductive system, we have accessory glands. Their function is to provide nourishment for the sperm cells, to provide support for sperm cells traveling through the female reproductive tract after sexual intercourse. The first one is called the seminal gland, otherwise known as seminal vesicles. It's located along the posterior surface of the bladder and produces the bulk of the seminal fluid. That's where we get the concept of semen, because most of the semen is going to be seminal fluid and sperm. Okay. Now, this seminal fluid is thick. It contains the sugar fructose, so that's going to be for energy. You're going to have certain proteins, citric acid, inorganic phosphorus, potassium, and prostaglandins. The prostaglandins are going to help with smooth muscle contractility, both in the male and to a point in the female reproductive system. Okay, and I may have gone a little bit off there. I'm sorry about that. Here you see this area, and now we get into the prostate gland. Now, the prostate gland is a chestnut-shaped structure located inferior to the urinary bladder, so it's below the urinary bladder. Muscles of the prostate gland also help propel the seminal fluid into the urethra during ejaculation. The prostatic fluid protects sperm, helping to live longer and be more mobile. Okay, so basically some of this fluid is starting to act on them, and the fluid will increase certain activity. The prostate gland gives off certain materials. These include enzymes, zinc, and citric acid. Finally, we have the bulbourethral gland, otherwise known as the Culper's gland. This is a pea-sized structure located inferior to the prostate gland. The fluid is secreted during sexual arousal, and it acts to neutralize the acidity of the urethra in preparation for the passage of the sperm cells. So basically, this small secretion will neutralize any acidity from any remaining amount of urine that's in that urethra beforehand, so it won't cause this. So if you look at this, you will see from a side view here, you have the urinary bladder here, ureters. You have the ductus deferens coming around and somewhat a little bit on the behind side. You have the ampulla here, and that's sort of the end of the ductus deferens, and then you have this mixing that occurs both from the seminal glands here, from their secretions, from the prostate and their secretions, and don't forget that you will have some addition here of the bulbourethra, but the bulbourethra comes a little bit beforehand, sort of to prepare the way for the proper movement of the semen without the harsh acidity of urine damaging the delicate sperm cells. Okay. Now, when we look at the external male genitalia, these include, of course, the penis and the scrotum. The scrotum, otherwise known as scrotal sac, is suspended inferiorly from the floor of the pelvic cavity. It is posterior to the penis and anterior to the anus. We have two muscles, the dartos muscle, which is a thin band of smooth muscle deep to the dermis of the skin. We have also a scrotal chamber, which is the site of the testes, and they're separated by connective tissue partitions called the scrotal septum. The cremaster muscle is deep to the dermis, thicker layer of skeletal muscle. Okay, so dartos is smooth muscle, cremaster is more skeletal muscle. It is an extension of the internal oblique muscle and is used to keep the testes temperature lower than that of the body core. Now, if basically the individual's temperature is much high, it will relax and allow the testes to be somewhat farther away from the body core. If it is much cooler out, it will almost reflexively create the situation where the testes, the scrotum will be brought closer to the body wall. Okay. And as you can see here, so we have here the muscles, we have here the scrotum, and you have the testes here. Okay. And we've shown you the basic background again. You can see here the cremaster muscle, and you can see here the thinner dartos muscle. Then also what you have is, of course, the separation that occurs right here. All right. Now, penile structures. The penis, it has several different structures. One of them is the root. It's attached to the pelvic bone and urogenital diaphragm. The body, which is also referred to as the shaft, is elongated cylindrical portion. The glans is the extended tip of the shaft. Now you have three major erectile tissue networks that exist in there. Two of them are the corpus cavernosum. The third is really the corpus sponginosum. So what we have are these vast networks of venous sinusoids and arteries. They're within a meshwork of connective tissue and smooth muscle. In essence, they provide the penis the erection that is necessary. When you look at the cavernosa, you have two dorsal columns at the root. They diverge to form the cruxa of the penis. The spongiosum is a single ventral column that encloses the penile urethra. And at the root, this chamber enlarges to form the bulb of the penis. Distally, this chamber expands to form the glans of the penis. Now, there's something called the prepuce, otherwise known as foreskin. It's a thin fold of skin that covers the glans of the penis. Now, the issue comes up about circumcision. Now, here's part of the reason. The foreskin on the inner surface will contain glands that secrete a waxy material called smegma. Now, the problem with this is it will provide favorable environment for bacterial infections. So a lot of times when little boys are born, they go through a removal, usually at birth, of the prepuce. Now, this can be done for cultural, religious reasons, as well as hygienic reasons. Some people have had objections to that. But what tends to occur is that there's been this removal of these cells. Oh, and by the way, there have been those who have used the foreskins and used them, the skin cells, to develop artificial skin, if that's of interest, okay? Now, you can see here's where the glans would be. The neck of the glans is right here. You have the body of the penis, and then, of course, you've got the root here. Now, if you notice up here, you have basically the two corpus cavernosa. Underneath it, and we'll go through a side view in a second, the corpus spongiosum, okay? And then, of course, you've got the external urethral orifice right there, okay? If you look through this side section here, this is what you have. You notice you're going to have the cavernosa on each side, and they will come to a root down here. You have the urethra coming through here, and this is the spongy urethra, as they would refer to it. It is surrounded by the corpus cavernosum, okay? Now, if the individual still has foreskin, this is how it would look. Notice that there's a small space there. Usually what happens is that it is surgically removed after birth, okay? Now, when you examine the microscopic anatomy of the male reproductive organs, we look at, examine the testes and epididymis. The testes contain seminiferous tubules. Okay, these are the tubules that you are going to have, and you've got to get used to this terminology, meiosis or meiosis. Some people, and this is basically the other means to cell division and replication, but it only occurs to make gametes. In other words, sperm cells, ovum cells. Why? Because when you deal with mitosis, you're making an exact copy of the cell. So it has, let's take humans, for example, 46 chromosomes. At the end of the division, you have 46 chromosomes. But in meiosis, you're going to have, starting with 46 chromosomes, but in the end, each and every reproductive cell will have only 23 chromosomes. Because when you take sperm and egg and brought them together, 23 and 23, you end up with 46 as a fertilized egg. We'll get more into this in development later on. Now, inside this collection of tubules, you will have 3 or 4 of them bundled together, making a lobule. And they're separated by connective tissue partitions. And so you have the seminiferous tubules, and inside the tubules, you will see basically a construction that will include the interstitial cells, otherwise known as Leydig cells. These are located between the seminiferous tubules surrounded by connective tissue. Leydig cells or interstitial cells produce the male sex hormone, androgens. Primarily, it's made of testosterone. Okay? Now, there's a lot here, but just run with this for a bit, and you can also review this in activity 31.4. When we talk about the term spermatogenesis, this is the development of sperm cells. Spermatogenic cells, these include all types of sperm cells in development. So the ones that are primary, secondary, spermatotids, etc. Now, spermatogonia are located at the base of the tubules. They are undifferentiated cells. They are continuously dividing by mitosis. Now, whoa, didn't you say meiosis? We'll get into that in a minute. Think of them as almost the stem cells to make more sperm cells. Once you start going into the status of primary spermatocytes, that's the initial differentiating of the sperm cells, and they will undergo meiosis. So they will start at basically 46 chromosomes and through a system of meiotic divisions. And the secondary spermatocytes are the end product. You will have now sperm cells that have 23 chromosomes of DNA. Okay? When you have spermatotids, these are sperm cells undergoing further maturation. Remember, you're going to have to lose a good amount of cytoplasm. You're going to have to sequester all the mitochondria into the neck. You're going to have to form a flagella, which is the transport medium. You're going to have to form a structure called the acrosome. The acrosome is necessary for the enzymatic burrowing in of the sperm cell into the egg cell. Okay, so there's a lot of biochemical and structural changes that occur. So when I say mature or formation, etc., there's a lot of stuff that goes on. Now, in this area, we have also nurse cells. They're also referred to as sustentacular cells. They extend from the base of the tubule to the lumen of the tubule and completely surround spermatogenic cells. Think of them as sort of the assembly sites and at the same time the nursemaid that will nourish the spermatogenic cells and regulate spermatogenesis. Sperm transport time through the epididymis is about 2 weeks. By the time they're ready to leave the epididymis, they're fully mature, ready to go. Now, tubules in the epididymis, these are sites of storage and maturation for sperm cells. Also, you will note in the epididymis are basically these structures called stereocilia. They're not really cilia per se, but they're elongated microvilli. Extending into the lumen of the epididymis, they function to absorb testicular fluid and provide nutrients to sperm cells. So they help, basically nurse along or manage the sperm cells as they're there. Now, there comes an interesting question. What if the person does not ejaculate? What if the person does not have intercourse or anything else? What happens? Do the cells get really, really, really old? No, after a period of time, I think it's around 64 days, the cells will be dying out and they will be reabsorbed. And of course, new cells are always being made. Okay? Now, the other interesting aspect is what we come up to is called the blood-testis barrier. This consists of the nurse cells with tight junctions. They basically divide the seminiferous tubules into the basal and luminal compartments. They isolate the immune system from developing sperm cells and their unique antigens. So if you take a look on the 31.4, you're going to see where these, if you can picture it, just sort of that round circle there, and nurse cells are on the edges. And basically what they do is they isolate from the blood and thereby from the immune system the differentiating and unique proteins that show up on sperm cells. Why is this so important? Well, here's where we go through a situation where it's an interesting question, and here's an interesting answer. What happens during a vasectomy? In a vasectomy, someone has a small section of the vas deferens clipped out. Usually what they do is they may also cauterize one end of the tube or both, depending. And what happens is this, that the sperm cannot successfully travel down the entire vas deferens. Now, what will travel down the vas deferens into the epididymis and into the testes will be immune cells. And it's sort of like, well, it's an unusual analogy, but pirates coming upon the treasure chest of gold. The immune cells come into the area where sperm cells have completed maturation. They have never met up with many of these antigens. And what happens with some vasectomies is some men are able to say, it didn't really bother me, I was a little sore. Others were saying I had to put ice on, I had to this, I had to take painkiller, etc. Because what's happening is they're having a massive immune reaction to the tissue and to the sperm cells that are unique in the epididymis and back down into the seminiferous tubules. So it can be sort of a situation of an immune reaction. Now, by the way, how does this really play a role for new and up and coming PA students and nursing students and pre-med? There's interesting studies that you will read, and they find that the reversal of a vasectomy declines profoundly as individuals have more time from the time they had the initial vasectomy. In other words, reversal would mean that they are able to restore their fertility. And unfortunately, within about a year, that gravely goes down that they will have success in a reversal. And within 5 years, forget it. Even if they do make sperm cells, they're coated with antibodies, and they're basically impossible to fertilize an egg. Moving on. Here you can see the rectate testes. Here you see the walls, and the seminiferous tubules are right here. And this is much closer where you see the seminiferous tubules. You'll see sort of the lumen area here where there may be some sperm cells occupying the centers here, some not. Here is a diagram that helps you a little bit better. What you see are the nurse cells around here, 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 and here. And the cells going through spermatogenesis are developing and eventually come to full maturation with their tails. Notice that they're no longer the round cells with lots of cytoplasm, but they're very small amount, basically almost a crystallized, tightly formed structure of DNA with the acrosomes, etc. Here you see the essence of that blood-testes barrier. And you can see that basically there is no way that the blood will go from a capillary or a small artery to get into exposure with the sperm that you see here. This is another image that I wanted to take time to review. You see the stereocilia here. These are the tubes of the epididymis. In the center, these masses here are sperm. You have some pseudostratified epithelium here in the tubes. You have principal cells here and here and here. You have connective tissue in between the tubules, and you have stereocilia, which are there to help mature these sperm cells that are ready to go. Okay, and this further image provides you with a clearer understanding of, here's the epididymis going all the way down here till basically the inferior part, and then it starts going into the ductus deferens. Okay, and this makes up what we call the spermatochord. Spermatochord, you notice that there's an artery and a vein, etc. And this is basically what you see. The testis separated by some of the structures here, the seminiferous tubules in these different lobules. You'll see the sperm in the lumen here. You'll see the stereocilia here. Okay. Examining the microscopic anatomy of male reproductive organs, the ductus deferens and the prostate gland. So we're just going to go over this briefly. And the ductus deferens, the walls are composed of smooth muscle. They will do the contractions, almost like peristalsis, to help move along the sperm. From the lumen going into the first inner lining, you have the mucous membrane, which has, of course, epithelium, which faces the lumen of the duct, and then underneath it, the connective tissue layer, which is the lamina propria. Then you have smooth muscle layers, which are thick and composed of 3 layers, longitudinal, circular, and longitudinal again. Finally, you have the adventitia, which is the outer layer of fibrous and loose connective tissue. Now you have a section sort of at the end of the ductus deferens called the ampulla. It's a dilated region, and part of it is so that you have some mixing of the different fluids coming in. The prostate gland is a glandular region separated by thick bands of smooth muscle and connective tissue. You have the prostatic urethra, which the glands secrete fluid into the urethra, as you can see right here. Okay, so you have the ductus deferens here, and you notice that you have the thick walls of smooth muscle. They're going to help propel the sperm down into the tubular area, and then you have, of course, the ampulla, and then you have this meeting where you're going to have, of course, the seminal gland, and you notice that it has a lumen, and there's lots and lots of secretory pockets for that fluid, and of course also there's smooth muscle on the outside to help squeeze and push more of the seminal fluid into this area here. Then you get into the prostate, which is connective tissue, smooth muscle, but also tubular alveolar glands. These are going to provide prostatic fluid and add to the mixture of semen. And finally, you have that small structure there of the bulbourethral gland. You can see, of course, the lumen. You have lots and lots of mucus glands, a little bit of smooth muscle, and then, of course, the capsule. Again, these are all glandular, adding different components to the mixture that's going to make up semen. When we finally get to the last section, which is really the microscopic anatomy of the male reproductive organs of both the penis and the spermatozoa. First off, let's talk about the penis. The erectile tissue, this is the dominant structural feature of the penis. We have the corpora cavernosa. There's a pair of them. They're deep. They have central arteries that are deep inside, and basically they have the blood traveling through each of the corpus cavernosa, and they will provide the main blood supply for the venous sinusoids. What happens is the artery delivers the blood. The venous parts will take away the blood in the erectile tissues. But what they do is they sort of trap the blood for a period of time. Okay? Now, there's also a central structure, and that is the corpora spongiosum, and that's where you have the penile urethra passing through it. By the way, a small minor issue. You may have heard propriism. Now, if you ever hear some of the Cialis or some of the other drugs for erectile dysfunction, Viagra, etc., they always say, if you have erections of more than 4 hours, please contact your doctor. All right, let me explain to you. Propriism, and it can be painful, is a situation where literally the blood is still in that penile area for an excessive amount of time. And this partly comes when individuals, really without their doctor's consent, are using Viagra. Sometimes they overdo it. Sometimes situations occur. And what will happen, curiously, is that they will end up with an erection lasting too long. The blood flow is not being allowed to, the sinusoids don't relax and don't transfer the blood back into the rest of the body. You don't have blood exchange. Literally, you can start having tissue dying inside. And that can be a medical emergency in some cases. Sometimes surgery is required. Okay. Now, I know some might be laughing at this, but it is a serious medical situation that has to be considered. That's why they say if you have an erection for more than 4 hours, you have to basically notify your doctor and perhaps come in for some treatment. Now, when we talk about this, we're going to talk about the tunica albuginea, which is a tough fibrous connective tissue sheath that surrounds the corpus cavernosa and the corpus spongiosum. Let me just jump ahead on that. So here is what you see. You have some dorsal artery veins and nerves here. You have the urethra here. So this section here is going to be the corpus spongiosum. This larger area here and this one here, this is the deep artery of the penis, and as the blood is flowing back, it's going to fill up these venous sinusoids. You have a dense network of elastic fibers here, and so this is how the erection will form. Now, when we talk about sperm structure, keep in mind, as I said before, sperm structure, sperm contain only half the DNA found in other cells of the body. They're haploid, okay? 23 chromosomes for humans, okay? So when you look at the structure of a sperm cell, you have the head, which contains the DNA, and right at the cap, at the tip, is a structure called the acrosome, and it provides enzymes to break down the outer protective layer of the ova, that's the egg cell. Then you have the middle piece and neck that consists of mitochondria to provide ATP for the movement of the tail, okay? And then, of course, you've got the flagellum, the tail. And you can see this all clearly laid out here. The neck is here. You have what sometimes they call the neck and the middle piece the same. But here you have this very long tail. It's a flagella. Now, notice the heads here. They're all basically either kind of rounded or kind of somewhat almost arrow tip shaped. There is a way to determine if you have defective sperm. One of them is literally if you have two flagella. In other words, there can be a flagella coming out the head. There can be a flagella 2 or 3 at the back end. Okay, these suggest that if they did fertilize the ovum cell, you might have a defect, gene defect. Okay, so what are the activities you're going to do today? Review the gross anatomy of the male reproductive system, including any model images. The model images that I do include will probably be from the book right now. Review the microanatomy of the testes and sperm development, including review the micrographs. So all the micrographs that I've shown you, you need to review. Complete the worksheet, which is pages 563 and 564. You need to upload the worksheets by Friday, 5 p.m. They are going to be in a PDF form. So all you have to have is something of a PDF reader, and you can insert the necessary information into those two sheets. Well, actually, one sheet, it's 2 sides, and then upload it. I also encourage you to review the lab videos and lab manual notes. And with that, I hope you have a great